to 5 prime.
Mm.
So then it would still be like this answer, except it would be reversed. It would be AUC GGG CAU, like that. So just make sure you read the question, and these ones are super important. Just write it out. Like have a piece of scratch paper with you. Write out the original answer. It's like write out this one first, and then just pair it right below it. And then even like if it asks you to flip it, Like as soon as you see it ask for that, just write it down. Because if you start right away and go for it, you'll take less than a minute on that question still, and you'll for sure get the right answer. Here we go. The nucleotide sequence in one strand of DNA is 5 prime. All this 3 prime with the nucleotide sequence of the complementary DNA strand. Same strategy again. A goes to T, A goes to T. So immediately we're down these two. T, A, and C because G, C right there. Nice. I don't think he'll get this mean. At one point, there was a question where he gave you the the amino acid sequence. It might have been on a quiz. I don't think it was on the test. We'll see if he asks one like this, where he would give you like the amino acid chain and to ask you what's the DNA sequence for this amino acid chain.
Ooh.
That was like super mean.
Yeah, that's nasty.
I don't think he did that on a test. But yeah, stuff like that just honestly at that point, if it's gonna take you that long, just click and move on and come back if you have time. Okay. I think we're starting to beat the horse with this one. This is super important. Remember the 3 prime, 5 prime. Look at these answer choices. More than likely, it's going to be one of the 5 prime ones. It can't have a U. Can't have a U. ATG. This is going to be TAC. TAC. Not that one. Not that one. Should be B. Yeah. Which, if you can do it that way without writing the whole thing out, power to you. There's very low stakes right now, so I don't really care. I can do it quick, but the recent COVID-19 pandemic, they love these questions. I guarantee you, like there's this question, this was like almost this exact question was a boards question as well. Since COVID, they have loved these COVID-19 questions. Same in microbiology and in microbiology boards, you'll have at least 1 or 2 questions about COVID-19 just because it's still so fresh. The reason COVID-19, okay, I'm just going to skip away. Identify the nucleotide base and the type of virus. So, I don't think we have to read the question. Genomic virus. Genomic analysis of this virus has shown the percentage of guanine and cytosine nucleotides G plus C content varying 32 to 43%. Scientists also found existence of a special nucleotide with no methyl group. Identify the nucleotide base and the type of virus. So, I don't know if there is like a technical way to like figure this out from the question, or it's just knowing memory. It's a uracil RNA virus. So COVID-19 is an RNA virus, but
when I was first going through these, I just remembered because when the shots first came out, the vaccines first came out, it was an mRNA thing, and so
yeah.
The only connection I can make to the lecture was back in chapter 16 when he was going through the different nucleotides, and he talked about uridine being different from thymine in that it doesn't have that methyl group, but
Yeah.
it's
So
still
that's
random.
probably the key right there, is that no methyl group. It's a lot of words to just know it's an RNA virus and call it a day. Yeah. You'll see a handful like this again. It's the same. I think it's the exact
I think it's
same
the
question.
exact same question.
Yeah. Yeah, uracil RNA virus. Like I said, this will pop back up a handful of times with COVID-19. There's a couple of like specific, you'll learn this in microbiology, pathology, and if you take the board prep courses, there are a handful of viruses, or not just viruses, but like infectious diseases that have like specific events tied to them. Like we talk about like anthrax, you talk about leprosy, you talk about cholera, and like there's very specific like case studies and specific like environment studies and stuff that like attached to these specific diseases, and they'll like ask specific things about like those specific cases. So you'll learn all of those as you go through more of the program. So the start signal for most organisms,
AUG.
AUG. The template strand of a segment of double-stranded DNA contains the following. What is the base sequence of the mRNA that can be transcribed from this strand? So from C, G, A, U, C, G, A, A, C. Sorry, ACU. There it is. C, don't do too fast. Otherwise, you mess up like I did. Just more of that back and forth and back and forth. Transcription is described as which of the following?
RNA synthesized
by DNA. So D.
by, yeah.
Yeah. DNA synthesizes our mRNA. What chemical bonding is present between the base pairs of DNA double helical structures?
Hydrogen.
Yeah, or hydrogen bonding. Same as our protein stuff. What do DNA polymerases require for replication to begin?
RNA primer.
Yeah, you gotta prime the thing before it can do its thing. What is one of the general functions of RNA polymerase?
Biosynthesis of RNA.
Yeah, we talked about that specifically which RNA polymerase is responsible for mRNA.
2.
Yeah. What is replication of DNA? I think we already talked about this. This is another repeat question. DNA from DNA. What is the function of amino acid tRNA synthetase? So breaking down a little bit real quick, synthetase. ACE means it's an enzyme, and synthetize, what does that mean? Synthesize.
You're making something.
Yeah, so we are making our tRNA, right?
Mm-hmm.
So we look at these answer choices. We're not forming RNA, mRNA. We're not generating amino acids. So it can't be all of the above. So bam. We are attaching the amino acid to the corresponding tRNA. Amino acyl tRNA is just the fancy way of saying tRNA with the amino acid attached to it. Most often, you'll just hear it said as tRNA. But the technical name for once it has the amino acid bound to it, then it's the aminoacyl tRNA. What is transcription? Another repeat.
Yeah, A.
Yeah, what is translation?
That's